Okay, so we're gonna go backwards through the ventricles. Back here is the fourth ventricle. This is a side view, and we saw earlier that's looking down on the fourth ventricle. The sheep, so that's looking down on it. That's the side view in the sheep. This is a side view in the human. And from there, we go to the cerebral aqueduct. The same thing. There's a cerebral aqueduct in the sheep. There it is in the human. Here is the third ventricle. It's a midline structure. Here it is in the sheep. Here it is in the human. Okay, so here's the third ventricle, and this hole right here, that hole goes over, and you can see the probe coming out on the other side, because
you can't see it.
You cannot see it? Okay. Hold on.
Oh, there you go.
Yep. See that? So I went through to the side. This hole is called the foramen Monro. It's spelled without an E, so M-O-N-R-O, Monro without an E. And it goes, there's a Y. So from the third ventricle, there's a Y that goes out to the left ventricle on either side. And that's what this is. This is, I've just sort of torn back the septum, and this is the this is the lateral ventricle. This thing that's overlying it, this is the corpus callosum, if you remember that. Let's get some of this septum out of the way. Okay, so we can kind of see what's going on here. All right. And what you notice is that there's this smooth cavity here, smoothly lined cavity, and that is the lateral ventricle. And then there's this crud in it, this thing, and this is the choroid plexus. This is the tissue that makes the CSF. So the CSF is just being churned out. We're making a lot. I can't quite remember how much, but on the order of a liter or so each day. So a lot. And it has to, so the pump is in the lateral ventricle, or the biggest pump is in the lateral ventricle, and it goes backwards through the foramen Monro to the third ventricle, back through the aqueduct, through the fourth ventricle, out Magendie and Luschka, and the two Luschkas, and then that surrounds the whole brain. And so any interruption, any blockage, if we block that third ventricle, which is really easy to close, if there's any pressure, if we block there, then the CSF backs up, and now there's too much CSF, the choroid plexus keeps pumping, there's no way to drain it, and that causes hydrocephalus. Okay, so now let's follow the lateral ventricle, and there's no good way to do this except for to sort of dissect it by tearing it. That,
see it perfectly.
Yeah, so you can see it goes all the way forward to this frontal pole. Do you see that? That's the end of it. So it's going all the way forward, but the really cool part is as we go backwards. And so we're gonna follow it as it goes backwards. Look at that. I mean, it's very pretty. I think it's very pretty. And you see it's diving down, but do you see that it dives down and it goes back? And so let's follow it as it goes back. Ooh, look at that. We're going back to the occipital pole. So there it is. There it is. The frontal pole, back through the body, back to the occipital pole, back here, and then it takes a turn and it goes down in there. Where's it going to? It's going to here. It's going to the temporal lobe. Do you guys see this? This is the C. The brain is very University of Chicago, and it has a C, just like the University of Chicago logo. So it comes back here, it has a backward spur before going down to the bottom of the C.
So it's a C off the midline a little.
It's a C that, yes, that goes around like a ram's horn. And so now let's try and follow it as it goes into the temporal lobe. And we're going to pick it up right here. Okay, look at that. There we are. There we are. There we are. And we see our friend, the hippocampus. So all I've done is I made one little cut so to reveal. I'm going to have to make a little bit more of a cut just so that you can see it. We'll take a little bit off of here. Okay. So, what do we see? We see this. We see this ventricle going all the way up to the pole. This is the front of the temporal pole. Do you guys remember? This is the back, this is the front, cerebellum, temporal lobe, and there is the lovely hippocampus. In the temporal pole of the lateral ventricle. So this lateral ventricle has gone like this. It sent a spur back here, and then it came all the way front here. So let's do one thing. Let's just take a look at this. Can you get that? Mm-hmm. So you can see this is back at the end of the occipital pole. This is where, right down under here is the hippocampus. This is the temporal pole of the lateral ventricle, and that was the frontal pole of the lateral ventricle. A nice C. Okay? And if you look down on it, you can see that it's not in plane. It comes out as it goes forward. Do you see that? It comes out as it goes forward. Shall we just look at that one more time because it's so pretty? Okay, one more time. Here is we're going forward to the front of the lateral ventricle, back, 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 to the back pole, which is really close to the occipital pole of the brain. So front to back, and then around to get that temporal lobe. Can't forget the temporal lobe. And there it is. There's the part, that's the end of the lateral ventricle in the temporal lobe. And all along the way, there's this stuff, this choroid plexus. So it's being churned out of the lateral ventricles. Churned out of it also gets produced in the third ventricle. It also gets produced in the fourth ventricle. It does not get produced in the very vulnerable cerebral aqueduct. You wouldn't want to put choroid plexus here because it would possibly clog this up, and then everyone would have hydrocephalus. So we don't want that. The same deal that we just saw in the human is also true in the sheep. And so we'll just quickly look at the sheep ventricles. Okay, so I cut far enough down, and there I've entered the lateral ventricle. I can kind of try and follow it. A little hard to do. The best tool for this, for you, those of you doing this at home, and I know at least one of you is a tongue depressor. Okay, so there you go. We're going back to the back end, and we're going to come up here. It's not a great dissection, but we're coming up there. There's the front end. But the most fun part is to try and get it into the temporal lobe, and so that we can see a nice hippocampus, we can see the nice C shape, and there it is. So there, look, I just went in, and there it is, and I can follow it.
Can we show it again? A little more so. Yeah. There we go.
Okay, so now we're going to go like that. Oh, look at that. It's so pretty. Ooh, yes, and there it is. Sheep and human, both mammals. There you go. And you see a little choroid plexus. This stuff. It's choroid plexus. It's a little less elaborate.
It has
There
less
it is.
surface area.
Has less surface area. So, but there it is, and there's the hippocampus.
Where would the amygdala be then?
And the amygdala is going to be right up here in front. And thank you, Sol, my very valued assistant.